Check 1 2. Check. Check 1 2. Check 1 2. Check. check 1 2 check. Check 1 2 check 1 2. Test 1 2 check 1 2 check 1 2.
Hello. That one's fine. Hello. Hello. Oh, what we could do? Oh, maybe we could do I don't know. We'll see the middle. You tell me anything. Yeah, there we go. So what we had did the last time, one is not working.
Check.
We sat halfway. Then if one takes that half and then one takes the back half and the front half because then it's easier to get to people. Yeah. Oh, Kiara's on that end there, so that section there at the end should be fine. She says she'll sit at that end of there, so maybe
Check,
the
check,
front section.
check. So I'm just gonna
Okay, so I was gonna sit
push
here,
it
but
a
while I'll
little
do it,
bit.
I'll place
Hello,
myself in the
hello,
middle so I can like
hello,
maybe do that section on that side there and some off the side. Yeah.
hello.
Okay, perfect. Thank you. Yes, one. I'll try and front and like. Okay, so you are this side, right? Yeah. Okay.
Morning, everyone. Um, just a few announcements before we start. Um, just that because of the air cons, when you guys come in and out, please close the door behind you so that we can keep it nice and cool on the inside. Um, and that speakers that want to be mic'd up, to please come up to the front to the AV guys if you want to have the hands-free mic, so that you can get mic'd up while the presenter before you was busy presenting. And then just that AC and aircon technicians will be working a bit, so hopefully there won't be too much noise. But if you do hear anything in the ceiling, it could be them. And then lastly, there's a bit of changes in the program now before tea. Firstly, in the first session, Ricardo Haldo is a poster, so that there's a gap there between Rachel Keane and Courtney Reed. So Courtney, come up to be mic'd up if you want when Rachel is already presenting. And then myself, Tertia, and Sivu's talk will be moved towards the end of the climate session for streaming purposes. But we are going to give people just a bit more time, so we're going to start in about 2 minutes, okay? And we're just going to call any of the last people inside. Thank you. Okay, guys, that was the bit of a delayed start we're giving everyone. We had to wait for our two general managers to get here. Okay, everyone, welcome to the soils session. From bedrock to biosphere, looking at geology and soil sustaining savannas. First up for our session, or should I re-announce the announcements? Maybe? No? Okay. You guys will catch up, the rest of you that came in late. First up, we have Gladness Koza, who will present on mapping soil nutrient distribution for forage quality assessments and grazing management in Kruger savannas. Thank you, Gladness.
Okay, good morning, everyone. My name is Gladness Koza. I'm a PhD student at the ARC. Yeah. Okay, I think it's still not showing here. Okay. Okay, so today I'll be presenting to you on soil nutrient and forage quality assessment in the Kruger National Park. Uh, okay. So the range length cover over 70% of the Earth land surface, and they support more than 2 billion people in the whole world. And they are critical for biodiversity conservation and ecosystem health. However, monitoring them can be difficult as they tend to be costly and time consuming. And so to keep this, remote sensing was introduced or adopted to help with the monitoring. And amongst these tools are the vegetation indices like the NDVI were introduced to help with this. And the NDVI, was then sort of adopted as the standard for monitoring the range length because it can mostly be used for biomass estimation, productivity, and vegetation health. And with this came the assumption that a high NDVI would mean a healthy range length. But with a system like the Kruger, for example, a healthy NDVI does not always translate to a healthy rangeland, because even unpalatable grasses or low nutrient ones can still appear to be green while they are of low nutrition. And so for this, we aimed to assess how soil nutrients affect or influence forage quality in these rangelands in the Kruger. Our study area was then in the central Kruger in the Satara, and we took along the H7. So I don't know, like maybe people who have been along that route may have passed there and saw that some of those places, they have wide open spaces, but usually there are no animals grazing there. Okay, so for our methodology, we did the soil sampling in the dry and wet season of 2024. And then from that soils, we took them to the lab for analysis of potassium, phosphorus, calcium, magnesium, and pH. And then at the same time, we flew the drone, which we used for the NDVI, to calculate the NDVI, both in the dry and the wet season. And then for our results for the soils in nutrients, we found that in the dry season, I can say like maybe things were rather stable. The nutrients, such as the potassium, calcium, and magnesium, they were fairly like stable, with only the phosphorus being low, and the pH was slightly acidic, which like usually it's more or less what grasses would prefer, where the grass are slightly acidic. But then now when we get to the wet season, we realize that there's a change. There were huge increases and decreases, especially we see like with the magnesium, it had a very big increase from what was originally there in the dry season. The phosphorus, it decreased, and this was something like it could be attributed to maybe it was being taken up or it leached off, because in the wet season, that's when the rain start. And the pH, it became neutral. And this, it all becomes a problem for the growth of grasses when now they are moving towards neutral, as they are not, most of them, for them to grow being of quality, the neutralness is not what they would prefer. And then we also did the NDVI. So we mapped the NDVI, and then that one on there on the left side, it's of the dry season. And then we can say like on the dry season, it's like, well, it's mostly dry, so there's not a lot going on. But in the wet season, see, like everything is green, and this shows that the grasses are growing there. And this shows that the grasses are growing even though the nutrients may not be corresponding very well with them. So we correlated the nutrients that we got with the NDVI, and we found that there's very little correlation or no negative correlation between the NDVI and the nutrients. But what we found interesting was the correlation between calcium and magnesium and phosphorus and magnesium, as these were high negative correlations. So this causes problems when magnesium increases a lot. When there's high magnesium, it becomes a problem for phosphorus to be taken up. Because in such a system where phosphorus is already deficient, it becomes even way much worse, because now it means the grasses, they will grow, but without the phosphorus. And then now another problem becomes the calcium and the magnesium. When they have a negative correlation, it means when one is increasing, the other is decreasing. So it means that now they are going to fight for exchange sites in the soil. And this also becomes a problem for the grazers, because when they are fighting like this, it means now it might as well be the same as when there are no nutrients, because it will cause the problem for the grasses. And while all of this is happening, the grasses will still grow because they are getting water and everything that is there. And the NDVI will still show that appear to be green, even though there'll be nutrient decoupling, and the grasses will be starved of the phosphorus that it needs to be nutritious. So now we say that our low phosphorus mean it highlights that there's a chronic nutrient deficient in the system, and this will limit the growth of high quality grasses. And this we have even noticed in the field that it led to the growth, like more dominating increased grasses of like the bedrock lower species, and they dominated both in the dry and the wet season because they are able to grow well in even in low soil phosphorus fertility grasses. And the problem with these grasses is that they produce high fibrous biomass, which also reflect as high in the NDVI, but it's of low quality. And now when you look at the NDVI, you would see that, okay, everything is very high, and then some might even overestimate the grazing capacity. But now the problem with that is that the grazing capacity only caters to the biomass. It's not looking at the nutrition that is underneath of feeding the grasses that are in that system. So in conclusion, we can say that the green grasses do not always indicate quality. Could just be forage quality, and just because grasses are there, it does not mean that they are of good quality. And this is sort of a case of looks can be deceiving. So it's what you get, what you see is not what you get in most of the cases. And for this reason, effective management is important so that it can integrate the above ground with the below ground, so that we can always have a balance between the quality and the quality of the forage. Thank you.
Ernest. Is there any questions? There's time for maybe one or two questions. Oh, here we go. Corley.
Thank you for your talk. As you're speaking, I had so many questions, and I won't ask all of them. I'll try and only ask one. But so you only showed us quite a small patch, and I was wondering, like, there's a lot of heterogeneity in the landscape. So you will also have sodic sites. You know, you will have different different places on the Katina.
Yes.
And animals move amongst those patches. And we also, we know that wildebeest, for instance, has this short migrations within the park.
Yes.
So I was just wondering, do you think that animals can balance out their nutritional needs by using the landscape cleverly? And can we pick it up if we look at sedentary animals versus animals that move around?
Yes, I think that could be possible, because now, and I think from some of our sites, we have noticed that there are animals that tend to stay there on most of the places, and they graze there, and then maybe in a different season, you find that they moved and in different places. But then now when it comes to maybe a system like this one, we know that the grasses that were dominated did not have a very good smell. So maybe thought that because the grasses that dominates there, I think they call it stinking grass, then they don't like that smell, so they don't go to that side. And now the grasses that smell good, they end up dominating. And since now we've been coming in different seasons, we've noticed that they are usually the first ones to grow. So even if the good grazing grasses grow, they grow late once those ones are already dominating, and maybe then the animals would just pass because they would see that and they don't eat. Okay.
Okay. Thanks, Gladness. We'll move over to our next speaker. Next up, we have Lena Naftal, who will be talking about mapping the distribution of soil organic matter in savannas in Namibia and Angola. Thank you so much, Lena.
Good morning, everyone. As I was introduced, my name is Lena Naftal. I am from the Namibia University of Science and Technology, and today I will be talking to you about the vertical distribution and driver of soil carbon in woodland ecosystems of Namibia and southern Angola. Sorry. Soils is a very important reservoir of soil carbon, as it stores more carbon than vegetation, which is up to 3,000 pentagram of carbon globally. The issue is that the sampling strategies in Southern Africa mostly focus on the carbon which is on the shallow depth, which is 30 centimeters above ground. And so as a result, very little is known about the deep soil carbon. And this is more especially for countries like Namibia and Angola. So this study aims to fill this research gap by looking at three objectives, which is to quantify the vertical distribution of soil carbon in 1 meter deep pits. And with this, we want to look at how carbon, how much carbon is missed if only 30 centimeter of the soil is sampled. We also want to relate the total soil carbon to above ground biomass carbon stock, as well as to identify the environmental drivers that are controlling the variability across sites and depth. This study was conducted at 5 study sites following an increasing gradient of rainfall, starting from Ongava Game Reserve with the rainfall amount of 400, and looking at 3 sites in the northeastern Namibia, which ranges in the rainfall between 500 and 700 millimeter, as well as Kruger National Park, which is in the range of 800 to 900 millimeter per year. These sites are in different vegetation types, back to Ongava in the Mopane woodland, the northeastern Namibia, in the northeastern Namibia with the in the Baikia Pterocarpus woodland, as well as Kruger National Park in the Myombo woodland. Soil samples were taken in 1 meter soil pits, which was sampled then at the depth of 0, 20, 40, 60, 80, and 100 centimeters. The total carbon was analyzed with the elemental analyzer, and bulk density was calculated using the oven-dried soil cores. We also gathered environmental drivers from different sources, such as ERA5, soil grid, and as well as SRTM. Looking at total soil carbon across sites, we can see that the carbon vary between sites, ranging from the dry. So looking at the graph on the right, starting with the sites with the driest rainfall to the sites with the wettest rainfall. So it shows that Ongava Game Reserve, there is a lot of variability, with Ongava having the high median, and as well as the high Yeah, okay, thank you. As I mentioned, Ongava has the high median as well as the high within site variability, and the sites in the middle that are receiving medium rainfall, having to store low carbon. Further, on the graph on the right, we decided to aggregate our soil depth to correlate to the depth that is mostly sampled in Southern Africa. So having the 0 to 30 centimeter, 30 to 60, and 60 to 100 as our subsoil, and as our deep soil, and we can see that the soil depth of 60 to 100 is storing high deep sock. Further, we calculated the proportion carbon to understand or to see how much percentage carbon is stored at each depth, and we can see that at least from 30 to 60% of carbon is stored below 30 centimeters. Most of the time, people are at a crossroad between sampling at a depth of 30 centimeter or to sample at a depth of 0 to 100 centimeter. So here we did a regression where we compared the carbon for the whole pit, that is on the x-axis, on the y-axis, and carbon at 30 centimeter on the x-axis. And here we can see that over 50% of this carbon is stored below the 30 centimeters. Carbon storage across these woodlands differ in carbon storage patterns, with some storing nearly half of their carbon below 60 centimeters. And so there is a difference between the sites where one of the two sites are storing their carbon in deep sand, in deep soil layer, and then we have 3 sites which are in deep sand that are having their carbon mostly evenly distributed. Further, we went on to compare how above ground carbon stock relates to soil carbon, and looking at this graph, we can see that above ground carbon stock is a poor predictor of deep carbon. Looking at the drivers of carbon allocation at different depths, we can see that for the topsoil layer, soil carbon is mostly driven by climate, while the soil carbon at depth is mostly driven by bulk density. To conclude, deep carbon represent a majority fraction of total soil carbon, and soil sampling only the 30 centimeter underestimate soil carbon stock. The driver of soil carbon differs between surface and deep layer, and different woodland ecosystem vary in storage patterns. Thank you very much for listening. I welcome your questions.
Thank you so much, Lena. Are there any questions? We have time for one. You see? Sorry, Sally. You see?
Yeah, thank you so much for your presentation and the interesting results. I was wondering, how did you assess above ground carbon stocks? You didn't say anything about that.
Thank you so much for your question. So for above ground carbon stock, we used measurements where we set up 1 hectare plots, and we measure all the trees using the diameter, and we calculated the above ground carbon stock using the allometric equations to derive the above ground carbon stock for woody biomass, woody species.
Okay. Thank you. And then one more question quickly by Sally.
Hi, Lena. Thank you for a really interesting study and important data. Your last graph showed that as above ground carbon stocks increase, then the soil organic carbon decreases, which is a really important message to tell people who are wanting to manipulate above ground carbon to try to store carbon, because if you're storing above ground carbon but losing below ground carbon, then that's like a problem. So were you expecting that? Were you surprised by the data? What are your thoughts about that?
Thank you for your question. Yes, indeed. Before I analyzed this, my thought process was always, or let me say, my hypothesis was that we should expect to find high below ground soil carbon in areas where you have high above ground carbon, because above ground should feed the soil. But after this, I was like very surprised the result, and now that changed my perspective of how I look at these two pools. Yeah.
Okay. Thank you so much, Lena. So we'll be moving over to our next speaker, who is Rachel Keane, and she'll be talking about bush encroachment and whether it's a boon or a barrier to groundwater recharge. Thank you, Rachel.
Hi, everyone. Yeah, I'm Rachel Keane. I'm based in the States, kind of with Oregon State University and Kansas State University. And I'm going to talk about a project that is very much in progress. So it'll be some preliminary data and then talking a little bit about our plans to continue sampling. And this project is in collaboration with some folks at SAON. And we're interested in thinking about bush encroachment and the spread of trees and shrubs in savannas and grasslands, and thinking about how that impacts water fluxes, especially in the subsurface. So there are a lot of ways that changes in vegetation cover can impact hydrology. Some are direct and some are indirect, and I won't cover all of them today. But the ones that we're kind of the most interested in thinking about are potential changes in water uptake and how that might shift zones of soil drying, especially in more mesic sites. When we get increased woody cover, that often is accompanied by increases in evapotranspiration, which can lead to kind of deepening zones of soil drying. But this isn't ubiquitous across sites, especially drier sites. We don't always see that trend. And then below ground, as we have these woody plants come in and have very different rooting systems to the grasses, the roots can really alter the subsurface structure of the soil. So we have the roots kind of physically moving soil particles around, but they're also potentially exuding different compounds that can impact the way that soil aggregates. And this can all influence pore structure and pore connectivity, which then can influence the way that water moves below ground. So we're also interested in kind of this downward vertical flux of water and seeing if this change in subsurface structure is accompanied by changes in infiltration, which ultimately has impacts on groundwater recharge and water yield. So our main questions in this project that I'm going to talk about today are first just looking at how woody encroachment or bush encroachment is impacting subsurface structures, that's roots and soil structure, and then how that might be influencing these water fluxes below ground. So wet-dry cycles at different time scales, and also just general infiltration dynamics and how much water is making it into deep soil layers. So for our study design, we have 4 sites. We wanted to capture a little bit of how climate can be impacting these trends as well. So we have 2 drier sites that are shown in yellow, where mean annual precipitation is less than about 650 millimeters per year. And then we have 2 wetter sites where precip is over about 800 millimeters per year. And at each of these sites, we set up woody grassy pits. So we have paired woody grassy locations at each of our sites. This is an example from Ntumkulu, which is it is our driest site. So that we can compare kind of woody grassy dynamics that are adjacent to each other at each of these locations. And so at each of those sites, we're looking at root dynamics. So I'm going to share some abundance data, but we're going to have biomass data coming as well. We're looking at soil physical properties. We installed soil moisture sensors to get hopefully several years of soil moisture records. And then I'll talk a little bit about some soil geophysical methods that we're going to start implementing next week, actually, when we go back out to our sites. So we'll first talk about kind of the structure of the subsurface and how that might change with bush encroachment. So we have, like I said, root biomass data coming that is not yet all picked and measured. But what we do have right now is root abundance. So when root biomass is kind of looking at how much root is at each depth, root abundance is more looking at what fraction of the soil contains roots of different size classes. So it's a kind of a complementary different view of thinking about root dynamics. And to do this, we, once the soil pit is dug, we clean off one face really well and take a high resolution image. And then, I don't know if you can see this, but we overlay a grid of 1 centimeter squared grid cells on top of the soil profile and actually go through and count presence absence of roots of different size classes. So this is an example from Maripskop, which is our wettest site. And it also has kind of the most dramatic comparison in root dynamics between the two, the grassy and woody sites. So these are surface on the top panels and then a deeper kind of chunk of the profile on the lower panels. And so this data gives us, at 1 centimeter depths through the soil profile, it gives us like a coarse root fraction. We also have total root fractions. But the coarse root fraction is kind of what we're interested in thinking about how these roots might influence water fluxes. So at Maripskop, you can see that there are very dramatic increases in coarse root abundance in the woody site compared to the grassy site, which is what we would expect to see. And especially those deeper layers where we have an increase in woody root abundance or coarse root abundance is where we're thinking that we might see impacts on water fluxes and especially into those deeper soil layers. So these are the profiles from all 4 sites, and you can see that Maripskop on the right is kind of our most dramatic comparison. But at all the profiles, there are chunks of the soil profile where we do see increased coarse root abundance in the woody plots compared to the grassy plots. So we're interested in thinking about kind of how these root dynamics will then impact water fluxes. We have only a little bit of soil structure data so far, but I wanted to share what we do have. So this is total porosity, which we got after collecting bulk density cores at different depths at each of our profiles. And total porosity it's the total pore space in the soil, and it's not directly correlated to like saturated hydraulic conductivity, but increases in porosity do kind of give the potential for increases in water flow if those pore spaces are connected. So it gives us kind of another clue into how the subsurface might be shifting. So at our dry sites, we don't see big grassy woody differences. But at our wetter sites, we start to see a divergence where the woody plots have higher total porosity than the grassy plots, especially again at that kind of woodiest site at Maripskop. So together with the higher coarse root abundance, this increase in porosity kind of gives us the hint that we might expect higher infiltration rates because we're opening up new channels of water for water movement in the soil in these woody plots. So now we'll talk a little bit about the water data that we're actually getting. We have soil moisture records that are just kind of starting to come in and get long enough for us to work with. So I'll show a couple of them today to just show the trends that we're seeing so far. This is Maripskop again, our wettest site. And you can see that the tree and grass plots are clearly different. And the woodier sites tend to have lower soil moisture across pretty much the whole record at this site. So at wetter sites like this is what we expect to see because year to year you get some soil water storage. So woody plants have extra water to take advantage of. At really dry sites, there may just not be enough water available for woody plants to take advantage of. So with these records, we're interested in looking at kind of the event scale wet-dry cycles. So how much infiltration, how much of a soil moisture spike you're getting after a rain event, but also how much dry down is occurring between rain events. So we also have data from one of our drier sites at Wits Rural, and we're also seeing lower soil moisture under the woody plots at Wits Rural as well at this drier site. So we have these records coming in and we're starting to see these overall trends, but we're excited to start digging into that event scale dynamics a little bit more in the future. So the last thing I wanted to talk about was some measurements that we're going to start making next week at all our sites. We're going to start measuring electrical resistivity, which is a soil geophysical method that complements soil moisture measurements from sensors really well. Whereas sensors, we get very high resolution data. It's from a very kind of discrete pocket of soil where that sensor is located. But electrical resistivity gives you a lower resolution look at a much broader region of the subsurface, and you can get deeper into the soil than is usually feasible with soil moisture sensors. So with this method, we set up a transect of electrodes, which are really just large metal nails, and run a current through that, which pushes electricity into the subsurface, and then we measure electrical resistivity of the soil. And we can use this as a proxy for soil moisture dynamics because increases in moisture are typically accompanied by decreases in electrical resistivity. And so we measure this at the beginning, like the wetter portion of the growing season, which is what we'll start doing next week. And we'll repeat these measurements during the end of the growing season when things are drier. And if you subtract those two, you can look at kind of a seasonal drying trend under woody and grassy areas. So we haven't done this yet at our sites. We'll get that data hopefully very soon. But I'm going to show an example from some work that we did, similar work in the US. So in this case, our major encroachers were shrubs, as you can see, and these transects span kind of grassy into woody, so you can see the transition really clearly. So more red areas indicate that more drying has occurred over that time span. So under the grassy areas, you can see that most of the drying is really concentrated to those surface soil layers where there is a lot of grass root biomass and where they're very competitive at water uptake. But under the shrubs, you can see that you start to get more of those deeper, kind of darker red zones where we're seeing increased soil drying down, a meter or 2 meters into the soil. So it complements our soil moisture data really well by giving us kind of a longer temporal scale and also a broader view of the subsurface and some of these moisture dynamics. There's the deeper zone of soil drying. So we're starting to see this trend of lower soil moisture at woody sites and are excited to dig more into some of those soil moisture dynamics. We're also going to, in the future, be looking at kind of the probability of preferential flow occurrence, which is that rapid movement of water deep into the soil through root channels and other macropores in the soil. And then ultimately, all of this data we're collecting, we're hoping will allow us to model groundwater recharge dynamics so that we can ultimately be looking at changes in groundwater recharge and water yield across these sites. And yeah, I'll thank the folks who have been helping us with fieldwork and digging a lot of deep soil pits and just for equipment support and kind of logistics at Oregon State. And I can take any questions.
Okay. Thanks so much, Rachel. Do we have any questions? It's time for maybe one quick question. Oh, sorry. I saw Carla first. Okay, Carla's giving it to Dave.
Hi, thanks, Rachel. I'm familiar with some of those sites, and they have different dominant woody species at the site. So I'm just wondering how much thought you need to put into the specifics of the species rather than lumping all the woodies together.
Yeah, the sites they differ quite a bit in dominant woody species. They also differ quite a bit in geology. So there is a lot of those kind of site context variables that we will have to consider. I think we'll mostly be looking at the paired comparisons within sites, but that was part of the reason that we wanted to get some more detailed root dynamics or like root measurements to see how, because like at Hannertsburg, for example, it's a lot more shrub encroachment so that we could expect the root dynamics to be different. We're also going to, when we set up these ERT transects, we're going to take plant surveys. So along those transects, we'll have like tree densities and tree species that are present to kind of factor that into our interpretation of the results. But it does differ quite a bit between the sites.
Cool. Thank you so much. We'll be heading over to our next talk now. Thank you, Rachel. So next up, we have a talk by Courtney Reed. Remember I mentioned earlier, for those that were here, that Ricardo Holders is a poster. Sorry about the error on the program. So Courtney is part of the Megacarcasses project team. So there's going to be 3 consecutive talks on the Megacarcasses project. So yesterday you had the crayfish crew. Today you have the Megacarcasses mob. So, just before she starts, I just want to draw your attention to a documentary that will be airing on this project tomorrow evening at 4:30 up until 5 o'clock. So it's a 20-minute documentary that will be basically documenting this project that we've been conducting in the park. It's called The Legacy of Mega Herbivores. So come and have a cinema type viewing tomorrow where we will be premiering the documentary tomorrow. Okay, so over to Courtney. She'll be talking about the nutrient legacies from elephant carcasses that drive cold spots of biodiversity and hotspots of ecosystem processes. Thank you, Courtney.
Thanks, Saoirse. And thanks everyone for being here. So today I'm really excited to share our research on the legacy effects of elephant megacarcasses here in Kruger. And I just want to start by thanking our collaborators on the megacarcass team, and also by highlighting Michelle, Zhao, and Helga, who are also going to be giving talks this week. And of course, none of the work we do would be possible without support from Sandparks, Scion, and the National Science Foundation. So we all know that animals play a really key role in nutrient cycling in savanna ecosystems. And one of the classic examples are termite mounds, which create these nitrogen-rich hotspots that attract herbivores like this elephant, who are now chowing down on these nitrogen-rich grasses. And we've also heard some talks this week on other nutrient hotspots like rhino middens and grazing lawns that are really created or maintained by animals and play key roles in nutrient cycling. And so animals also continue to contribute to nutrient cycling after they die. So I took this in Chaquan. This is a wildebeest carcass that isn't going to last very long. And so nutrients from carcasses, they move into soil or they're consumed by scavengers and dispersed. And we often think of them as pretty short-lived resources because of that dispersal or consumption. And so, yeah, they've always just kind of been assumed to be these kind of ephemeral resources in ecosystems. But part of the reason behind that assumption is because there hasn't really been, to date, that much research on megacarcasses and the role that they may play in terrestrial ecosystem ecology. So we know that megafauna, like elephants, have these really outsized impacts on ecosystems during their lives, but we don't know how those ecosystem engineering impacts may continue after they die. And that's actually really important because an average adult elephant carcass has more than 2,000 kilograms of carbon, 300 kilograms of nitrogen, and 125 kilograms of phosphorus that are all being deposited into this one very localized portion of the savanna. And so not knowing where those nutrients go or how they continue to impact the ecosystem is a really huge gap in our understanding of terrestrial ecology. So with this project, we're asking what are the legacy effects of elephant megacarcasses on above and below ground biodiversity and ecosystem processes. And to answer this question, we sampled 40 elephant carcass sites across Kruger, ranging from about 8 days to about 8 years post-mortem, and split across the granitic and basaltic soils. Today, I've just lumped the soil results for simplicity, but Zhao later is going to get into some of those specific nuances across the granitic and basaltic soil types. So at each of these sites, we collected soil and grass samples at the center of the carcass site, and then 15 meters away from the center as our off-carcass control distance. And we analyzed those for nutrient content. Then we also performed vegetation and grazing surveys at those on-carcass and off-carcass distances. And what we found first is that elephant carcasses release huge quantities of nitrogen into savanna soils. So just to orient you to these plots, so x-axis is carcass age, and then the y for most of the plots you look at today is going to be the log response ratio of on-carcass versus off-carcass. So anything above the dotted line is going to mean that this is enhanced on carcass sites, and anything below, it would be suppressed. And what's really cool here is not just that ammonium and nitrate levels are really absurdly high. So for ammonium, actually up to about 3,000 times greater on carcass compared to off carcass, but also that the effect lasts a long time. So 3 to 4 years for soil ammonium and the full 8 years for nitrate. And one of the major impacts of these nutrient inputs is a decrease in microbial diversity. So we did 16S sequencing for soil bacterial communities, and we found that bacterial diversity was suppressed at carcass sites, and it was also very strongly negatively correlated with soil ammonium. And we found the exact same thing with fungal diversity when we did ITS sequencing of fungal communities. And what we found was suppressed diversity for the first several years, and again, a very strong negative correlation between fungal diversity and soil ammonium levels. And there's actually some really cool fungal compositional changes here, and Michelle is going to get into those in her talk in a minute. Another thing to note here is that we actually ran these same correlation tests with soil nitrate and soil phosphorus and didn't get significant results. So it does seem that these decreases in diversity are really driven by the soil ammonium. So we ran the same test with plant diversity. And this is another really cool one where you really see that this effect is lasting the full 8 years post-mortem that we've measured so far. A really strong plant diversity suppression. And again, we see negative correlations, this time both with soil ammonium and with soil nitrate. So together, what we've seen so far is that the nitrogen inputs from megacarcasses have really strong suppressive effects on both below ground and above ground biodiversity that in some cases can last for a full 8 years post-mortem. So we've looked at biodiversity, let's look at ecosystem processes. Below ground, soil microbial respiration is strongly elevated in carcass sites and carcass soils, and it remains so for more than a year. So that response is relatively short-lived compared to some of the other variables we've looked at, but it shows that these soil microbes are responding really strongly to that initial large nutrient input. And above ground, we find that grazing intensity is strongly elevated at carcass sites and is actually still continuing to increase, indicating that there might be some kind of like positive feedbacks and ongoing processes kind of driving those patterns. And that's likely driven by attraction to high nitrogen in grasses at carcass site, which remain really highly elevated during the full period. And we see that grazing intensity and grass foliar nitrogen are strongly positively correlated as well, which kind of further undergirds that mechanism. So altogether, these results are demonstrating that megacarcasses generate these integrated ecological legacies that last for at least 8 years post-mortem, resulting in these long-term cold spots of biodiversity, but hot spots of ecosystem processes. Thank you.
Thanks so much, Courtney. We do have time for questions, if there are any. There we go. Okay.
Thanks for that, Courtney. Just very interested how you were measuring diversity, because diversity could be a measure of relative abundance and not necessarily species richness. So I'm just curious to see, is one species, whether it's the microbial or the fungal dominating, which decreases diversity, or is it just a decrease in absolute diversity?
So we measured Shannon diversity, and we checked evenness as well, which I didn't show here, but we find decreases in species evenness. So it does seem that some plants are responding, are like colonizing more quickly and in greater abundances. So there's like a pretty large plant compositional change. Helga is actually going to give a talk on Thursday that gets into that in much more detail.
And then at the back we have Ricardo. Oh, okay.
Thank you very much. That's very interesting stuff. But I'm wondering also about, so did you do any work on like the species that end up being left over, what that trait shifts are, and whether you can see the trait shift reflecting the nutrient or the trampling disturbance response, or like which are the species that survive this?
Yeah, that's a great question. So kind of above ground, really what you get is mostly a total wipeout of the plants that are there. So it's less, I think, about who survives and more about who's colonizing afterwards. So we get like pretty intense decreases in like plant biomass that then recover over time. And you tend to see early colonizers being things that like grow and reproduce quite quickly. So we see Urochloa trichopis and Megathrysis maximus are typically kind of the first grasses that are coming in. So those, yeah, fast-growing grasses are really the first things.
Okay. Thank you. Yeah. Oh, Judith has a question.
Thanks for that. So I'm just interested to know, on your x-axis you had your carcass age up to 8 years. How did you measure that?
Yeah, Sandparks keeps coordinates and dates for the elephant, dates of death, and so they were able to provide those for us.
Okay, thank you. Thanks so much, Courtney. Next up, we have Michelle Budney, who will be presenting on behalf of Nate Lemoyne. And hers is basically looking at how megacarcasses shape biogeochemical processes in Kruger. Thanks, Michelle.
Hi. Thanks, Tertia. So as Tertia said, Nate Lemoyne was originally going to give this talk. I'm his lab manager at Marquette University in Milwaukee, Wisconsin in the United States. We found out Saturday Nate wasn't going to be able to make it, so this has been a short preparation time for me, but I'm going to do my best. So as Courtney went over a really comprehensive explanation of our megacarcass project, and today I'm just going to go over the snapshot of how those megacarcasses change the soil fungal communities at these sites. So to review again, an elephant dies, it decomposes, and it deposits hundreds of kilograms of nutrients into the soil. So we had 40 sites across the park that we took soil samples at and ran nutrient and microbial analyses for. So Courtney showed this too, but we'll go over it again. When we looked at the nutrient analysis, it seems to have been nitrogen seems to be a really big driver in a lot of the changes that we see. So in this figure, the top portion shows that nitrogen in the form of ammonium and nitrate were both elevated on carcass sites versus off carcass sites. The bottom part shows the log response ratio. So just a reminder that 0 line would indicate that there's no change between on and off carcass sites. But we see elevated ammonium very early on after death at these carcass sites, and that decreases but stays elevated even 8 years later. The nitrate stays elevated at a pretty consistent level across that time. But we think that could be due to nitrification, the ammonium being converted into nitrate. So Courtney went over a lot of the great ways that changes the ecosystem. So I'm gonna get into one really close snapshot, and that's the fungal community. So the same samples that we took to get the nutrient data from, we extracted DNA and ran quantitative PCR as well as sending it for Illumina sequencing of the ITS region, which is the ITS gene region is a very common marker used for identifying fungal communities. So all the way across the park, we identified at all of our sites a total of over 16,000 fungal species in the soil. But if we zoom in to just the carcass site level, there were about, on average, 100 species found on the carcass site and about on average 200 species found 15 meters away off the carcass site. So it's a pretty obvious suppression in diversity on and off. But doing quantitative PCR, the ITS copy number, so on this figure, again, the x-axis is our years post-death, and the y-axis is our fungal ITS copy number, and that can be used as a proxy for community abundance. And we saw that on versus off, there was no change in abundance, even though we saw lower diversity. So this kind of suggests that the remaining species in the soil increase in abundance to kind of compensate for that reduction in species diversity. So to kind of go over that change too. We looked at the relative abundance of, or relative dissimilarity of on-carcass communities versus off-carcass communities. Very early on, so to orient you to this graph again, the y-axis is dissimilarity, wood 1, meaning that the communities are completely different from each other. So right after the death, we see that they're almost totally different, and that change does decrease, but even 8 years later, there's still only about a 10% overlap between the on-carcass and off-carcass community composition. So then we look to see where these changes might occur in the community. So to shift it into looking at a couple functional groups, the first one would be the saprotrophs. So those are your decomposers in the soil. They take the plant and animal matter and convert it into nutrients. We see that, again, there's an early increase on carcass sites. Again, we're back to our log response ratio. There's an early increase in decomposers that persists throughout that timeline, and which kind of makes sense. You have a large decomposing body, and decomposers are going to increase. So we also looked at the symbiotic fungi, which are they're plant mutualists, so mostly mycorrhizae that are connected to the plant roots. And we see a very sharp decline in these symbionts, and it stays pretty decreased throughout time. We think that there's a couple different reasons that it could be. So one thing Courtney said, that we have a lower plant diversity, so just lower plants in general, or we can go back to our nitrogen story. And as nitrate increases, we see that, so nitrate levels are on the x-axis, and are relative abundant of plant fungus symbionts on the y-axis. So these graphs are split out into soil type. The kind of brighter line shows our on-carcass communities, where the lighter line is our off-carcass. In both sites, we see as nitrate increases, we see a decrease in plant symbionts. It's more pronounced that suppression is more pronounced off carcass, and it's more dramatic on the basalt soils, which that soil type we're still trying to tease apart. So that's really two big takeaways, is that that there's a big disruption either between there being no plants, or this increase in nitrogen just means the plants don't need these symbiotic fungi. So our key takeaways that we've found: species richness of soil fungus species decreases on carcass sites, but their abundance does not change overall. The community composition does change and stays changed. Our decomposers increase, and the symbiotic fungi decrease. So thank you all. Big thanks to our collaborators who've worked on this project, and to Sandparks. Again, please come see our documentary. I will answer what questions I can, and if not, you can direct them to Nate Lemoyne at his email address.
Cool. Thank you. So does that mean you can't answer questions?
I can answer. I might be able to answer
Because
questions.
there's time.
I'll try.
There's time. We have a question by Kate at the back.
Thank you very much for your talk. So I'm really interested that, you found these changes on these carcasses, such as the change in fungal composition. But I imagine these elephant carcasses are quite few and far between. So to what extent do you find these effects on smaller carcasses, other animals, but perhaps for shorter periods of time?
I don't personally have any experience with smaller carcasses yet. That's a great question. I would think there might be some literature out there.
Okay. But
Courtney
we can
might-
also try helping you.
Yeah, what's that? She can't help me. Yeah, so I don't know how that compares to smaller sites is the answer.
Yeah.
Thank you.
Maybe to help answer that as part of the team. The difficulty though is that smaller carcasses, they spread out way quicker than an elephant carcass. You'll be surprised. Sometimes there'll be a carcass where the rangers have said that's maybe, I don't know, like 2 years old, 3 years old, and you go there and you sometimes struggle to actually find it. It's amazing how strong a lot of the hyenas and lions are to actually move that bones and the carcass apart. So the smaller carcass, that process might very well be at play, but it's so difficult to actually find the smaller carcasses out in the field. Yeah. Okay. Is that it? Anyone? Any other questions? Dave. Dave is probably going to answer as well.
Yeah, I was just going to answer. So prior to this study, the largest carcasses that have been investigated are those of elk and moose. So we're talking about animals that weigh 8 times what has been previously studied. So the effects that we're finding are sort of beyond what we have known previously.
Yeah. Thank you. Okay, so I think we can move on. There's no other questions. Thank you so much, Michelle. Next up, we have João Silva. He's also part of the megacarcass mob. And he'll be talking about parent soil and how it mediates spatio-temporal nutrient enrichment at the megacarcass sites. Thank you, João.
Good morning, everyone. Thank you very much for your attention. So I'm branching a little from this topic. It's my honor to be part of this group and of this study. So we were looking a little further into the effects of distance and time over different groups of nutrients that come from these megacarcasses. So yes, from until what we have now, the carcasses, they make a pulse of nutrients in the place they fall. And this pulse, this decreases with distance as expected. As you move further from the carcass, you would expect to have less influence from it. But something that is jumping to our eyes is that the different soil types, they make a lot of difference in the processes of these nutrients and everything involved with these carcasses. Also, we need to know how it works over time, as it was just asked now. How this process, for how long does this process last, and in which distance, and for which different nutrients, so the complexity of the whole question increases. The situation is a bit less romantic, as you can see in this picture, but still very interesting. When we get two different soil types to investigate, a nutrient-rich one and a nutrient-poor one, in these different distances from the carcasses, in different times, in a different time series, we can look at different nutrients and group them differently regarding their composition, because we expect they would behave differently due to their own composition. So if you get nitrogen, phosphorus, and sulfur-based nutrients, you place them differently from mineral nutrients and trace elements, so you can see different dynamics between and within these groups. In a model where we try and evaluate distance, time, and the interaction between them for each of these groups, okay? So when we look at these NPS nutrients, we can see that only the distance is the factor driving what we expect and what we observe from distances closer to the carcass to further from it. So you see, this is a multivariate data cloud, and you see the black dots as the central points, and you see clearly that higher quantities or higher concentrations of these nutrients here, very close to the carcass, and they fall and become pretty much similar as you move away from it. You have nitrogen from nitrates and phosphorus from this P-Bray method extraction, driving these differences, driving this pattern, and you see that they are indeed much closer to the carcasses than as you move further from it. When it comes to the nutrient-poor granite soil type, you see that yes, the distance and the time interact over this distribution of nutrients. So you can see here in this graph, also a multivariate data cloud, the breaking down into distances and the influence of time on the different distances. And you see that yes, for the closer distances, you have an influence of time. So the nutrients, they decrease in concentration in these distances over time. But that's not the same for as you move on from the carcasses. And again, you have phosphate and nitrogen as the main nutrients in this group, driving these differences, but they come from different parental compounds now, which is probably related to solubility in this different soil type. When we move on to mineral nutrients in the basalt-rich, nutrient-rich basalt, you see that yes, you have the distance following pretty much the same trend as before, with more mineral nutrients closer to the carcasses and decreasing as you move away from it. Also, an influence of the time, but regardless of the distance evaluated. So the bulk of the samples, regardless their distance, they tend to decrease in terms of mineral nutrients over time. When it comes to the nutrient-poor granite soil, you see again an interaction. So as time goes on in different distances, you will see a decrease of nutrients, of mineral nutrients, until the distance of 5 meters from the carcass. After that, this relationship is non-existent anymore. When we move on to trace elements, you see that there is no effect at all of the distance or the time over this group. Well, it's pretty much expected since we are dealing with very non-carcass dependent elements, right? So we're talking about trace elements that are found in very small quantities, and it's more likely that this is pretty much from the soil itself than from the carcasses leach or leakage. Okay, so we don't see a clear pattern of the distribution of these elements in the nutrient-rich soil over time or over distance. For the poor nutrient granite, we see that yes, we have an influence of the time regardless of the distance, which also kind of pushes us away from the influence of the carcass, right? You just have a decrease of these elements over time, so it doesn't mean that it's because or not the presence of the carcass. So again, these trace elements are harder to determine their dynamics and related to the presence of the carcasses. But in terms of effect size, we can see that yes, distance is by far the main factor dominating this distribution and these changes in concentrations. Time also has an influence on it, but always follow it by, always behind distance, and a much smaller contribution or effect of this interaction. So sometimes you see decreases in certain distances, sometimes not. But overall, in the granite soil, you have a much more sensitive situation or sensitive status towards these factors. So the granite soil, it suffers more or is more vulnerable to these distance and time factors that we propose to investigate. This especially for the NPS and mineral nutrients, not so much for the trace elements as we saw. So as conclusions, we can understand that nutrient concentrations, they change mainly due to distances, right? This is pretty much the takeaway messages. NPS and mineral nutrients, but not the trace elements, They behave very similarly in both types of soils, with highest concentrations near to the carcasses, and sometimes also in the shortest period. Nitrogen and phosphorus are the main drivers of both nutrient-rich and nutrient-poor, even though they come from different parental compounds, and this reveals a bit of the complexity of this interaction, soil types and the nutrients. And this has obviously consequences to all the trophic levels coming from it, right? So there is a community consequence for that. Different soil types have different main minerals and nutrient elements, also showing us that, okay, soil types are really important in this case. Nutrient-poor granite soil type was more sensitive to the distance and time variations, also pointing us that soil types are very important here, which lead us to ask why. Why is that? Exactly, I don't know. So we are looking deeper into that. My guess is as good as any. I like to think it's an informed opinion. I think the basaltic nutrient-rich soil will present much more organic matter. So obviously you're gonna have much more microbial activity, and this will kind of act as a buffer in the sense of the effects of the distance and other factors, time and climate factors or whatever. So maybe that's the way we must pursue, to look at the nature of these complex interactions and situations in the nutrient-rich soil, so we can understand better how these dynamics work. Thank you very much. I'm open for questions.
Thank you so much, Xiao. We have time for maybe, okay, I see 2 hands, 2 questions. We can start with Corley and then we'll head over to Lisa.
Thank you. So this is a question for you, but also for the other speakers. When we have in our Mediterranean systems that is very nutrient poor, when we have plants that invade, that increases the nitrogen in the soil, our fynbos, our Mediterranean vegetation that's adapted to that nutrient poor soils lose their competitive edge. So I'm wondering if the patterns that you see, is that driven by the high nitrogen and phosphorus, or is it actually just plants that deal well with disturbance that's managing to hang out in those patches? And then if we think of our basalt versus granite soils, like, do we maybe expect more disturbance-driven traits to persist on the one soil type versus plants that can actually deal with high nitrogen, high phosphorus?
So thank you for your question. If I understood it correctly, the influence of that on plants, right? So we have a talk on Thursday by Helga, where you see that the plant diversity decreases a lot close to the carcasses. Apparently the levels of nitrogen that come from the carcasses seem to be quite toxic to plant establishment. And as we just saw for the fungi, we also have loss of diversity. So we have loss of diversity of primary producers as well. And you have, which doesn't mean that they disappear, you have specialists there that are able to deal, especially some grass species, if I'm not mistaken, that can deal with these high levels of nitrogen. And the effects of that, like we saw, they last over time, but especially over distance. It's quite limited. It's quite well delimited, where you see the transition from these tolerant plants to more overall tolerant other species. I hope I answered your question.
Yeah. Thanks, Xiao. Over to Elizabeth.
Thank you very much. That's very interesting. I'm not sure if my question is well formulated, and I apologize if it's not, but I'm thinking about it again in a stoichiometric sense. Like, if now you've shown all of these minerals almost as one package, right? Minerals decline. But I guess there could be quite some specific predictions about which minerals, if we think about what the exact mismatch in stoichiometry is between the animals eating and the plants responding to an animal-provided packet of nutrients, right? So you would expect that as you have animals responding to this and coming in and kind of having this feedback loop that you guys were talking about, maybe there's additional nitrogen and phosphorus constantly being put back into the system through fecal matter and urine and these kinds of things. But there's certain elements that form the body of the dead animal that is now the sudden input. And those are the elements that are going to be systematically removed in the bodies of the new animals eating there, right? So I wonder if, do you think that there could be some predictions made about which are the elements that you would expect to decline more strongly based on that mismatch in stoichiometry?
Well, thank you for your question, observation. Yes, this is a very focused part of the whole story, right? So you have a whole ecological process going, species coming and going, different ones in different stages of the decayment of the carcass. So they also bring their own input of material to the scenery. But to try and answer your question, the main elements in this multivariate treatment, they really jump out to the eye. Like you can really see, okay, these are the one guys that are driving this data here and there all along. So it's pretty much those we showed, and we have to keep looking throughout all the plant analysis and all the other organisms involved to see if they're following the trend in everyone. But apparently it's pretty much clear that those that get highlighted, they have a huge influence in the data cloud, which means that they are the ones varying up and down through these factors we suggested. So yes, I hope I helped.
Thanks so much, Xiao.
Thank you very much.
Thank you. Okay. Okay, before we end the session, I just want to draw your attention to 3 posters in the session. One by Yossi Bada looking at soil moisture time series for various stations across the country. We've been monitoring soil moisture since 2019. Another poster by Tsumbedzo Ramaleva looking at soil property extremes and how it determines plant community and below ground bud banks in the Satara area, if I believe. And then lastly, a poster by Ricardo Haldo looking at independent responses of savanna trees and grasses to soil moisture and vapor pressure deficit. But that's the end of our session. Can we just give everyone a last round of applause? Thank you.
Thanks, Tasha. Morning, colleagues. I'm Guna Kensani, and I'll be chairing the climate change session. Just to remind everyone about the slight change in the flow of this session. The first 2 talks that are on the program by Tasha and Sivuyisiwe will be moved to the end of the session. So if Sarah can come closer, she will be our first speaker. And she is going to be talking about the effect of temperature on activity rhythm in a predator-prey system. Thanks, Sarah.
Hi, everyone. So I'm Sarah. I'm a French PhD student. And today I'm going to switch the topic and talk about large mammals' behaviors and the effect of climate change and temperature changes. So we all know that temperature are increasing worldwide, and that's also the case in African savannas. And you have here the prediction of African savannas temperature in the coming years. And this increase of temperature is known to impact wildlife. So it can have direct impact through physiological mechanisms, but it can also have indirect impact through behavioral thermoregulations. So behavioral thermoregulations include all the behaviors that an animal can do to cope with temperature. So when the temperature is very high, easy thing that you all know is that animal will look for cooler microclimate. So they will go into the shade in savannas, for example. but they can also change their activity rhythms, and this would be like resting more during the hottest hour of the day. And this is the behavior that we'll be focusing on today. For herbivores, there are quite a lot of studies that look at that, and but we can still wonder, is this really happening for herbivores? Because well, they just cannot rest forever when it's hot. They still have to forage. So they have to face a trade-off between the foraging requirements and their need to thermoregulate. So we are still wondering about that. And then for the carnivores, well, there is not that many studies that looked at carnivores and the effect of temperature on carnivores. And the few studies focused on diurnal predators and saying that they will switch their hunting efforts by the night. But we don't know anything about nocturnal predators. And for example, what are hyenas and lions going to do when it's hot during the night? So in this study, I focused on wildebeest, zebras, lions, and hyenas. And I just tried to answer this simple question: what's the effect of temperature on their behavioral time budgets? And the expectation is that the active behavior is less likely to happen when it's hot. We also tried to answer other questions like, are herbivores going to compensate their foraging during following nights? Or is temperature affecting the interaction between prey and predators? But I'm not going to talk about these topics because of time constraint, but I'm happy to discuss during the meeting if you fancy. So we use GPS location from collars deployed on these 4 species in Schusslo Infolocy since 2018. And we combine this GPS location with temperature data from global models of temperature that gave us hourly day and night ambient temperatures. And in this talk, I'm going to talk about temperature differences from the seasonal average. And anytime I'm going to talk about the behavior, it will be the behavior of the average temperature compared to the behavior that animals do when it's hotter than usual, so plus 7 degrees, and the behavior they do when it's cooler than usual, minus 7 degrees. But before talking about temperature, we need to change this GPS location into behaviors. And we used very well-known methods, that is hidden Markov models, that allow to translate this GPS location here and just transform them into behavior, where you have here in yellow the resting behavior, in blue the foraging behavior, and in green the commuting behavior. So now we know what are these species doing, and we can look at the probability to be doing this behavior according to temperature. So we used generalized additive models to do that, where we just estimate the probability to be doing the behavior according to the time of the day. So how does this vary throughout the day, and the temperature. We included interaction effect and random effect here. And the good thing with this model is that it allows to have nonlinear relationship between the variables. So that's the good point here. So if I just go to the results, we will have plots like this. So for each of the 4 species, we have the time of the day on the x-axis and the probability to be doing the behavior on the y-axis. So you will have different colors: yellow for resting, blue for foraging, and green for commuting. And for each color, you will have 3 lines, 3 different lines. One is the behavior that animals do when it's average temperature, and another one for cooler than usual and hotter than usual. So just as an example, for wildebeest here, if we don't care about the line type for now, we see that overall, when it's the hottest hour of the day here, beginning of the afternoon, wildebeest are increasing the time spent resting in yellow at the expense of foraging. So that's the expected behavior. And now if we look at the different curves, we see the solid line, it's what they're doing when the temperature is average during the season. And here the dashed line is when it's hotter than usual. So we see that when it's hotter than usual, wildebeest are increasing the time spent resting and foraging a bit less. So again, it's expected. But what is interesting here is that when it's cooler than usual, the dotted line, Wildebeest are increasing a lot their time spent foraging and resting much less, as if they were like making the most of this time period. If now we look at zebras, overall they are doing a bit of the same, like they are resting more during the hottest hour of the day. But if we look at differences between temperature, there is actually no differences. Whatever the temperatures are always behaving the same here. Now for lions, first here we see that throughout the night there is no big variation into behaviors. Then if we look at the dashed line here, we see that most of the night they are resting, even when it's hotter than usual. When it's hotter than usual, they are not changing their behavior. However, what you see right now is that the dotted line, this one, sorry, when it's cooler than usual, lions are reducing the time spent resting and increasing a lot the time spent searching for food. So again, making the most of these cool nights to increase their movements. And finally, for hyenas, well, we have a bit more of confidence interval here because we don't have that many hyenas colored. But what we see here is that hyenas, when it's hotter than usual, they are resting more during the beginning of the night, so they stop their activity later in the night. And then overall, throughout the night, they are spending much more time commuting, moving long distance when it's cooler than usual. So again, when it's cooler than usual, they are moving more. So that's the main result I wanted to show you. And just as a quick additional result, I just wanted to focus on the effect of absolute temperature, because since now I'm showing you results from the wet season, the hot wet season. But as a quick result, I wanted to show you for lions, if we put the temperature here on the x-axis and just look at differences of behavior for this temperature in different seasons, you will see that for lions, for the same absolute temperature, the behavioral response are not the same depending if it's the wet or the dry season. So I don't want you to focus on what are the lions doing, but just telling you that, well, it's not just about absolute temperature, but also about the context of this temperature and whether it has been hot since long before or not. But if I come back to the main question of this talk, what is the effect of temperature on their behavior? Well, we can say that high temperature is reducing the time spent active for most of the species we've been showing. But interestingly, this adjustment is more pronounced for cooler than hotter temperature, which was not that expected, actually. And here we are thinking about a scenario of ghost of past temperature-induced behavioral adjustments, where we can say that temperature in HIP has already increased. So maybe the behavior that we are seeing now at average temperature is already an adaptation to this past increase of temperature, and maybe they cannot increase their adjustment anymore, even if the temperature keeps raising. So that's just an hypothesis. And then overall, we found strong heterogeneity in our results, so different adjustments in terms of behaviors. And I think this study overall just invalidates all the blanket statements we can do about temperature effects on this species, and just a lot of heterogeneity as always. And yeah, I thank you for listening and happy to have questions and discuss more.
Thanks, Sarah. We have time for 2 questions. This one at the back there.
Thank you very much. That was super interesting. Do you know whether the animals are moving to shadier places? So those wildebeest, are they moving to places with more trees, for example? And that's one way that they're maybe able to keep foraging, but to regulate their temperature?
Yeah, obviously, as I was saying, they have different strategy to cope with temperature, so they can be moving to find like cooler microclimate and look for shade and these things. So right now we don't have accurate data of the vegetation in the park, so we cannot run this analysis where we want to know maybe they are moving, but maybe they are looking for more cooler climate, this kind of thing. So we don't have this information right now. That's part of the project and part of the aim we wanna reach. Thank you.
Hi, thanks. Hi, thanks. I just wanted to know, so on your plots, you plotted the herbivore activity from 6 a.m. to 6 p.m. and then your carnivore 6 p.m. through the night to 6 a.m.
Wait, can you repeat because I can't hear anything actually.
For your plots, you had the herbivores plotted from 6 a.m. to 6 p.m., the activity, and the carnivores you plotted the opposite through the night. Is it possible that the carnivores are becoming active earlier than 6 p.m., starting their hunting earlier on certain temperature days, or the herbivores are remaining active longer on those hot days?
Okay, I'm not sure I'm understanding the question, but you're talking about the 6 a.m., 6 p.m., and what's the effect on the herbivores? Things like this.
Yeah, I'm just wondering, are the animals maybe not treating that as a hard boundary? Maybe the herbivores are feeding later and the carnivores are...
Oh, yeah, sure. So that's the compensatory behavior of herbivores. Yeah, so we've been looking at that. So we looked at the behaviors of the herbivores during the night, depending on the temperature on the day before, to see if it has been hot during the days, they reduce the foraging, do they increase the foraging later in the night? So we've been looking at that. I did not show it because I did not have time, but what we found is that neither wildebeest, neither zebras are changing these behaviors. So whatever the temperature the day before, they are not increasing the time spent foraging the night after. Sorry,
Okay, thanks, Sarah.
Thank you.
We're now gonna move on to Neha, and she's gonna be talking about the diversity linked to tree clustering and rainfall in sub-Saharan Africa.
Hi everyone, I'm Neha Mohan Babu. I'm a postdoc at Utrecht University, and I will be talking about our work on diversity and spatial patterning in sub-Saharan Africa. As you can see, this is a big collaborative work with the CSO network, and I'm very grateful to all the data owners for sharing their data for this analysis. Savannas are inherently patterned landscapes because they are mosaics of grasses and woody plant cover, and this sort of patterning can go from more structured, spotted or striped kind of pattern to more unstructured clustering or even clustering of trees at the interface of forest-savanna boundaries. And like I said, there's a wide definition of spatial patterning, but for this talk, I'm going to be talking about any sort of clustering of trees as a metric of spatial patterning. And the reason we're interested in this is because patterning can actually make, recent theory suggests that patterning can actually make ecosystems more resilient to changes, and especially via community reassembly. And this makes it important to potentially understand the effects of spatial patterning on diversity in savannas. So we already know from the various talks we've heard and a large body of literature that savanna diversity is affected by a wide range of drivers. For instance, previous work suggests that along a rainfall gradient, savanna plant diversity increases. Fires and herbivory also have a huge impact, but they tend to be a little more nonlinear depending on the intensity and frequency of fire or herbivory. So we wondered if spatial patterning is also an important but understudied driver of diversity in savannas. There are several ways through which spatial patterning can actually influence diversity, and it could also depend on the mechanisms that cause spatial patterning. So I will go through three sets of hypotheses that we considered. The first one being scale-dependent feedbacks, where plant processes can actually localize certain resources, oftentimes moisture, which allows for facilitation at short distances, but causes competition at long distances. This difference in positive and negative in the distance of positive and negative feedbacks can actually result in patterning of vegetation. If this is one of the main drivers of pattern formation, we can expect that along a rainfall gradient, regardless of patterning, first of all, we do see that along a rainfall gradient, diversity should increase, but if if it's from a pattern plot, so to speak, we can expect that diversity is going to be higher at dry sites because pattern states, as I mentioned, would increase moisture in local patches, which would support species that would otherwise not exist in a dry state. But along the wetter ends of the spectrum, we can expect that moisture is no longer important or can even increase competition, thus reducing the difference between diversity in a patterned and non-patterned states. In contrast to that, patterns can also be an outcome of environmental templates. For instance, lots of savannas have termite mounds or nitrogen-enriching species, which can create localized increase in nutrients. If this is one of the main drivers, we can expect that diversity would be higher compared to that of a random state across the rainfall gradient. However, we can also see environmental templates that are stressful, such as like high salt stress or a low resource condition, in which case we predicted that diversity would reduce across the rainfall gradient. In contrast to both of these, patterns can also just be an outcome of stochastic mortality events. And especially if the frequency and the intensity of such events are low, we predicted that diversity might not be very different between patterned and unpatterned states. Given these wide range of possibilities and how patterned and unpatterned states could differ in their diversity, we are interested in understanding if spatial patterning and tree diversity are indeed linked to each other in sub-Saharan Africa. As I mentioned, we leverage data from the CSO network, and thankfully we have data from a wide range of environmental gradients. We used only the 1 hectare plots, and each of these plots, all adult trees are marked, and we have the XY locations of each of the trees, allowing us to quantify patterning. To give you a quick example, both of these plots are from the Gorongosa National Park in Mozambique, and the plot on the left shows a distribution of trees, which is more or less randomly distributed, whereas the plot on the right shows some clustering of trees. The way we quantified patterning is using Clark-Savins index, which is a metric that's based on mean nearest neighbor distances, and is compared to a statistically random distribution. And if the and we can classify plots as either randomly distributed, clustered, or regular distribution. In the CSO plot data that we had, we did not find any regularly spaced plots, so we just compared the diversity between randomly distributed trees and clustered trees. I will be showing you results from 3 different metrics of diversity, and in all cases, mean annual precipitation is the x-axis, and the diversity metric is on the y-axis. Moving to the first set of results is about species richness, and as expected, we see that species richness increases along a rainfall gradient. But what was interesting is that the green, which is the clustered plots that show clustering, had lower diversity at drier sites, but tended to have higher diversity at wetter sites compared to that of random plots with randomly distributed trees. This was also true when we considered other metrics of diversity, such as Shannon's entropy and inverse Simpson diversity, both of which also account for abundances of different species composition. Furthermore, we also tested for these patterns at various scales. So we tested it at 10 by 10 meter scale, 25 by 25 meter, and 50 by 50 meter scale for the same 3 metrics of diversity. And there's a lot going on this graph, but the main thing I want to point out is the fact that the green line compared to the black line is almost always lower at the dry end of the spectrum, and the difference between the two decreases at higher rainfall sites. So to summarize the results so far, what we find is that clustering of trees reduces diversity at dry sites, but increases diversity at wetter sites compared to that of plots with randomly distributed trees. However, unfortunately, this was actually not in congruence with any of the hypotheses that we considered initially, based on some of the mechanisms that we know are important for spatial pattern formation. One possibility, of course, is the fact that the processes that were causing spatial patterns in these very different sites are very different, and therefore we might not see a consistent trend. However, I do want to point out that we do find a consistent, statistically significant effect of patterning on diversity, which then begs the question as to what else might be happening. One possibility is that clustering actually does divide up the space, leading to some sort of patch dynamics that might be affecting species diversity. So for instance, if clustering sort of divides up the space into many small clusters, each cluster can act as its own island and can have very different species composition, thus increasing the species diversity at the plot scale. Similarly, a larger cluster can also have more number of individuals that increases the statistical chance of including new species. So we wondered if both cluster size and cluster numbers were actually changing along a rainfall gradient. We did see that number of clusters increased along a rainfall gradient for the plots that showed some sort of clustering. And we also saw that size of clusters increased with rainfall for the plots that show clustering. Taken together, this suggests that fewer small clusters are actually decreasing diversity at the drier end of the spectrum, whereas larger, more number of clusters are actually causing wetter sites to have more diverse plots. To summarize, we found that spatial patterning likely influences alpha diversity of trees in savannas. It also interacts with an existing environmental gradient, in this case rainfall, to suppress diversity at dry sites but increase it at wet sites. However, we only considered adult trees, so this analysis needs to be sort of extended to other size classes of trees and even non-woody vegetation, as it might be relevant for conserving and managing biodiversity in savannas. With that, I would like to thank all our funding sources and the CSO collaborators again, and thank you all for listening. I'll take any questions.
Thanks, Nea. We do have time for 2 questions. Sally?
Thank you, Neha. That was really interesting. I was totally with you all along the way. And you disproved your hypothesis, which I think is some of the funnest part of science. But then I was thinking, when you were getting at your mechanisms, isn't a key thing you want to measure is whether the clusters actually have different species from the matrix? Because you started to set up a bunch of new predictions that don't really make sense to me until we know whether the clusters are distinct communities within another matrix or just groups of trees that exist in a spread out way as well.
Great question, and I do agree with that. So the clusters are right now basically trees that were closer together than some of the other trees based on density-based metrics. But we did look at the nearest neighbors, and we do find that in some of the drier sites, they tend to be more likely to be monospecific strands, so which might also suggest some sort of dispersal limitation, but clusters in some of the wetter sites tend to be more diverse as well.
Okay, do we have another
I've got
hand?
the mic already.
Okay.
Thanks. Hi, can you hear me?
Oh, yes.
Yeah. Thanks for your talk. I might have missed it, but did you only look at rainfall or did you look at substrate as well? So the interaction with rainfall and soil, for example.
I looked at a bunch of different metrics, but in the end, rainfall was the only one that showed some sort of a trend in this data set. So for this talk, I just decided to focus on the rainfall trend.
But you didn't do any interaction, rainfall with soil, for example?
No, I did not check that, but that's a good point. That's something I can go back and look at. Thanks.
Okay, thanks, Nea.
Thank you.
Our next speaker is Laura, and she's going to be looking at demographic history of African wild dogs, reveals the role of savanna refugia in shaping diversity. Thanks.
Thank you for the introduction. There's one word I didn't put in the title, and that is genetic diversity. So you are in luck today. I'll be talking about that a little bit. So we know that savanna is a very dynamic biome that is structured by climatic and biotic disturbances. So normally these punctuated wet and dry cycles create temporal variability and a mosaic of different habitats. And we also know that the savanna biome supports an immense amount of biodiversity, including complex assemblages of herbivores and carnivores. So in the past, there have been repeated contractions and expansions of forest and savanna habitats during glacial and interglacial periods. So typically during cooler and drier periods, the deserts as well as arid grasslands expanded, typically at the cost of tropical savanna. So in many places, tropical savanna retreated in what we call Pleistocene refugia. So wildlife that was dependent on these tropical savanna often got isolated for long periods of times, and this is the main reason that we see a lot of interesting phylogenetic or biogeographic patterns across the range of species. So just to put this in a bit of a visual, so during different glacial periods, these tropical savannas contracted and expanded, like I mentioned, and what you then typically see is that populations start slowly differentiating across space and time, and this is due to three main evolutionary processes. So one is mutation. They will differ between populations, but it is an incredibly slow process. So this would take hundreds of thousands of years to see substantial genetic differences. What is a much stronger evolutionary force is genetic drift. So this is the random loss or fixation of alleles, which tend to differ between populations if there's no gene flow between them. And then you also have selection. This is the only process that's non-neutral. And it's basically, it doesn't create diversity, but it works with the diversity it has. So if a mutation is beneficial, it will increase in frequency. If it's harmful, it will decrease in frequency until it's removed from the population. So this can create differences between populations too, but you would only expect this if there's different selective forces at play, such as different environments or habitat. So typically, as a result of these phylogenetic forces, you start to see, if you make a phylogenetic tree of your species of choice, you'll start to see these different lineages or phylogenetic clusters, and most often these are still remnants of these Pleistocene refugia. So if populations have been separated long enough, they'll form a cluster. If they are locally adapted or separated for longer periods of time, they might even evolve into different species. So what's also interesting, if you look at the shape of your phylogenetic tree, it can tell you something about demographic history as well. Long branch lengths are normally from expanding population. Very short branch lengths are typically a species in decline. And sometimes you see very constant phylogenetic tree. This normally happens when you look at neutral regions of the genome where there's no selection at play. So don't get afraid. There's a lot more to it. We're not going to go into detail today, luckily, but I've been talking about demographic history. Obviously, there's a lot of confounding factors. You have, like I said, sometimes you have gene flow between Pleistocene refugia, sometimes you do not. It really matters what ancestral genetic diversity is shared between populations. There's background selection, divergent selection, and there might even be differences in mutation rate. So it's a complex story. But we're going to be talking about something a lot more fun, and that is the African wild dog, which is a charismatic savanna inhabitant with a critical conservation status. We know that it suffered tremendous demographic losses in recent years. And as a result thereof, the overall genomic health has become a central theme in conservation efforts. So today we're going to ask ourselves two questions. How are genetic lineages diverged across space and time, and how did demographic history shape genetic diversity? So we got some samples, 30 in total. We got 10 African wild dog samples from private reserves in South Africa. So these originated from reintroductions, and they're constantly being moved around to mimic gene flow. We've got 10 individuals from Kruger National Park. That doesn't need further explanation in this audience. And we've got some individuals from free-roaming African wild dogs in northern South Africa and Savi Valley Conservancy in southern Zimbabwe. So what we did to get a full picture is sequence the whole genome. And what we then did, you have all these genomes that you map to a reference genome, which in our case is an individual from Kenya. And what you then can do is see exactly where mutations occurred, if they differ between populations, whether they are in coding regions, outside coding regions, whether they occur in homozygosity or heterozygosity. But what you can also sometimes see is parts of the genome that don't have mutations or genetic variation at all. And this is what we call a run of homozygosity, and this is a direct effect of inbreeding. Typically, the longer your run of homozygosity, the more recent inbreeding is. So this can also then tell you something about the demographic history. So we first did a phylogenetic comparison. In this case, it's the mitochondrial DNA. And if you recall, you can see very short branch length, which is typical for a species in decline. It actually means that there's hardly any variation on the mitochondrial DNA. Most individuals are either similar or very much alike. You can see that this is similar to the figure in the middle, where you can see the effective population size decreasing quite sharply at about 0.25 years ago. Interestingly, this is also when the number of lineages increased, which you can see on the left figure. And this coincides with these Pleistocene refugia. In the phylogenetic tree, you can see that one individual from Kenya, and we measured that they diverged from South African wild dogs about 0.25 million years ago, which is similar to the other graphs, and this seems to coincide with tropical savanna habitat retreating during mid-Pleistocene climatic shifts. Okay, we also used the whole genome to compare effective population size through time. So in the graph on the bottom right, we start about 500,000 years ago. When we measured, there was approximately effective population size of 40,000 individuals, and they started gradually declining. So this is a period that is known to have very unstable had a very unstable climate, and you can see this gradual decline of African wild dog populations. Then we did another analysis to look at more recent changes in effective population size, and it seems that from about 400,000 years ago, the population was pretty stable until about 1,000 to 800 years ago, and there's a population crash. We call that a population bottleneck, and this seems to coincide with the human expansion. Then we also looked at genetic diversity. So nucleotide diversity is something that you can measure across the genome. It's a pretty good indication of genetic diversity. What first stands out is that nucleotide diversity is very low. This is about 1/10 of what you normally see in carnivores. What also stands out is that the managed metapopulation has a substantially higher genetic diversity, most likely as an effect of being sourced from different founder populations, and they're constantly trying to mix these individuals around to maintain genetic diversity. So in the middle, this is runs of homozygosity. If you recall, that's part of the genome where we don't find variation at all. Long runs of homozygosity is a sign of recent inbreeding, and we see the longest runs of homozygosity in Kruger National Park. We see a lot of short runs of homozygosity in the free roaming populations, which is indicative of more of inbreeding that is further in the past, and the shortest runs of homozygosity, so the least signs of inbreeding, is in the managed metapopulation. Then we also looked at genetic load. So this is what we call harmful mutations that occur in coding regions and that occur in homozygosity, which means they are expressed. In coding regions, it's most likely to have a harmful effect on the genetic health of an individual. It tends to have a phenotypic effect or might cause diseases or illnesses. So we call this genetic load, harmful mutations. And what really stands out is that Kruger National Park has a substantially higher genetic load, which coincides with the high levels of inbreeding we found. So in conclusion, we found that wild dog populations were historically large in Southern Africa, approximately 40,000 effective individuals. We found a gradual decline between 500 and 100,000 years ago, coinciding with Pleistocene climatic shifts. We found a sharp population collapse about 1,000 years ago, which we think coincides with human expansion. We found low genetic diversity, most likely as a result of these past and present population bottlenecks. Kruger National Park had the highest genetic load, so a large extent of harmful mutations and long runs of homozygosity, which are both indicative of inbreeding. And we found the highest genetic diversity in private reserves, which clearly emphasizes this positive effect that population admixture can play. We have 1 recent publications out and 2 in the pipeline, so stay tuned. And I want to thank everyone in the right corner for supporting our research.
Okay. Thanks, Laura. We have time for 1 question. Sam, and then keep it short, Sam.
Thank you, Laura. You know, there's always a challenge because what ecologists understand what a population is and what geneticists do are 2 different things. But I was wondering, what would the consequences be if I'm maximizing the survival of all wild dogs through my management actions? What would the consequences be on those indices that you're seeing there?
Yeah, that's a very complex question because there's a lot of depth to it. So there's a lot of proof that you don't lose these unique lineages. So wild ducks, there's not a whole lot to begin with, but a lot of people feel if there's unique lineages across your landscape, you shouldn't be mixing them because you might lose this unique genetic diversity. But now there seems to be an outcry that a lot of populations are stuck in small reserves, clearly suffering the impact of inbreeding. So sometimes maximum outbreeding is what you want. With endangered species, I think the risk of inbreeding always outweighs the risk of outbreeding or losing lineages. And like I said, there's evidence that you're not going to lose your unique lineages or local adaptation if you allow a small influx of individuals. So that would probably be my management. Oh, I'm misunderstanding the question completely. Okay.
No, that is very clear what you're suggesting
okay.
there. What I'm saying is I'm putting everything in my possible means in place to make sure every wild dog survives, including those ones with harmful genes. With the harmful genes, whatever lineage is sitting out here. So that's what I'm asking. If our management interventions in the short term don't have this effect, but what does it mean in the long term? If I have actually reduced natural mortality, in other words, selective pressures on individuals, which are what the consequences then would be.
Yeah, so that's like called relaxed selection, I think, where you take away some natural selective drivers in the wild. I'm a strong supporter of evolution. I think these natural processes will recover if you give animals the means to be able to adapt, which is genetic diversity. So if you allow a strong, large effective population size, perhaps with relaxed selection, maybe they haven't been exposed to lions or whatever other threats, I think these are processes that nature will eventually solve itself. Yeah.
Okay, great. Let's catch Laura during the breaks. We're gonna go back to the 2 talks. To our online audience, we're gonna see you after tea. So we're gonna head to Tasha, who's gonna be talking about the reverse revenues and relief of the recent Kruger floods.